Ideal Fertility IVF and Genetic Center, or this is our 35th journal club, and this is with two topics. One is an introduction to RASopathy, ciliopathy and tubulinopathy. Will be by Dr. Devashish. He will appear soon, and he will be talking on two case discussions. And with this RASopathy, ciliopathy and tubulinopathy, what is actually these pathies in fetus, and many of these abnormalities, the babies, they get born, and sometimes the things are so difficult in early neonatal life, in early adult life, or even in adult life. So as a sonologist or as a prenatal diagnostic person, it is our duty to identify these cases as early as possible, as far as possible, to identify these cases of RASopathy, ciliopathy and tubulinopathy. Actually, these are the group of the disease which are dependent on those genes of our body which are conserved throughout the evolution. Now, I will start this topic with ciliopathy. What is ciliopathy? Ciliopathy are complex which results in defective and dysfunction of cilia of the human body. Now, the question is, why we are talking about cilia? Are all cells are ciliated? Then what is the role of cilia? Can we see cilia in every cell? These are very important organelle in cells which are very, very important in embryonic life. Impact of defective cilia, defective and dysfunctional functioning in motile and non-motile cilia results in cilia motility and signaling defect that causes a large number of symptoms and disorders which are significant impact on those affected. As cilia are found on most cell types, defective cilia may affect multiple systems in body, causing blindness, defects, deafness, chronic respiratory infection, kidney disease, heart disease, infertility, obesity, and these symptoms have significant impact on affected. Some are devastating, most are life-threatening. Now, I will make a diagram to make you understand why I am talking about this ciliopathy disorder and why Dr. Devashish is going to discuss about the two ciliopathy disorder today. I am also having one case with me with ciliopathy. So, any cell in the body is having in interface a centriole. This centriole gives a tubulin sort of thing which makes a sensor on the cell. This sensor is just antenna of any instrument, any organism, like cockroaches, we see antennas. With this antenna, that organism can understand what is present in surrounding. In the same way, every cell of the body is having either a cilia which is motile or immotile. But there is a sort of antenna over the cell. Whenever there are other cells along with, it is also having an antenna. These cells, they talk with each other, and these antenna, they identify the activity of the other cell so that it affects its orientation, orientation of the surrounding cell, Each and every cell are sensing each other with these antenna so that they can distribute and function. Suppose it is a tubule of seminiferous tubule. It is having multiple cells. It starts from a single cell and this tubule forms because of there is a proliferation in a proper fashion so that it makes a tubule. So it makes a cavity and it makes a surrounding cells. Now each and every cell is having an antenna. This is called cilia. And these cilia, they interact with each other so that it can arrange itself in such a way that it makes a tubule. Now, if this antenna is sensing something, that means it is secreting some protein. And if this antenna is being made through this centriole and this dynein arm, then it must be having some gene. So there are multiple genes which are affecting this formation and function of each antenna or each cilia. Now, if any gene of this cell is mutated, abnormal, then this gene will not create a proper protein, so there will be defect in antenna, defect in cilia. If cilia are dead, then this tubule might become like, there might be gross collection of cells. They cannot arrange in such a way that they can make a tubule. So they will not make a tubule. So there will be formation of cell growth or no tubule formation in kidney. That means this antenna or this cilia of each cell at the time of embryonic activation or gastrulation or at the time of 3 germ layer, where there is the ectoderm or hypoplast, it makes gastrulation into the mesoderm. If there is problem with the cilia, ciliary protein or gene in the cilia, it will not happen. So there will be problem in the mesodermal function, problem might be in the endodermal function. So whole system get abnormal. And if this baby forms, then multiple organs will be affected because of this ciliary dysfunction. Now, there are 2 types of cilia in body. Few cavities like respiratory cavity or intestinal, they contain some cells which are having cilia moving. So these are motile cilia. They move in such a way that it gives a flow of the mucus, flow of secretion to a certain fashion. If there is a problem with the ciliary function, again there will be problem. So there are 2 types of cilia, which is one is immotile and one is motile. That means there are 2 types of cells which are having, one type is having ciliary, which is moving cilia, and another having immotile cilia. But every cell is having one cilia as an antenna to sense the surrounding. And this is a conserved gene. Conserved means in our evolution, if we started from paramecium, some single amoeba, then go the nematodes, arthropods, then mammal. These ciliary cells are present from here. Then only there will be proper distribution of the cell and proper arrangement of the cell, proper left and right sidedness of the cells, left or right sided of the body. The heart will form on the left side. The liver will form on the right side. Why not liver forms on the The while left lobe of the liver goes shrinked and right lobe gets dominated. Why the heart goes to the left? So if there is ciliary problem, there will be problem in the left and right sidedness. So there will be heterotaxia. So there are multiple problems with multiple organs affected by the ciliary dysfunction. Now this ciliary dysfunction is again structurally, each cilium comprises of microtubule backbone, the ciliary axoneme surrounded by plasma membrane, and I will show the figure. Motile cilia are characterized by typical 9+2 architecture with 9 outer microtubule doublets and central part of the microtubule present as multiple motile cilia on cell surface. Non-motile primary cilia with 9+0 architecture appear typically as single appendage. So this is the basic structure of cilia. This is the basal body and the cilia forms and it projects from the cell surface like this. This is the ciliary membrane and these are the microtubule pair 9+0, here 9+2, the motile cilium. Now, what is the impact of defective cilia? Defective and dysfunctional functioning in motile and non-motile cilia results in ciliary motility and signaling defect that cause a large number of symptoms and disorders which have significant impact of those affected. Why we are reading in this way? Because there are multiple defects in the fetus. We can make them as a syndromic sort of thing, and now it has been discovered there are a lot of defective genes. They might present in a different manner in different organ, but they appear together. So cilia are found most cell type. Defective cilia may affect multiple systems in body, causing blindness, deafness, chronic respiratory dysfunction, and these are different type of diseases in the neonate or in early childhood which may create a problem, and most ciliopathy genes are autosomal recessive. Why it is so important to study all these things? Now suppose one baby is abnormal, then there will be another abnormal baby in the same family because it is autosomal recessive. That means the husband and wife are carriers. So you can make it out how important to diagnose that fetus which is showing abnormality in ultrasound, and we just do an DNC and evacuation without knowing the genetic status of the baby, of the fetus. So it is very important whenever we find something which is syndromic in ciliopathy sort of thing, always think about that there will be a problem with the husband and wife. They might be carrier of this because they are autosomal recessive. There will be more 25% chance of having another baby with the same disorder. So ciliopathies are a different name, and if Devashish comes, he will display a case of Bardet beidle syndrome, Alstrom syndrome. These are different syndrome with multiple problem in a single baby in a particular fashion. So this is a Bardet beidle and nephrolithiasis, polycystic kidney. I will present a polycystic kidney patient. Husband is having autosomal dominant polycystic kidney disorder and they want an IVF because he is having azoospermia also, because there is a problem with the vas difference, because there are multiple organs affected. The husband is having multiple cysts in his kidneys with high blood pressure, with raised creatinine, and want to create baby. Last is retinitis pigmentosa also. These things, all ciliopathies, because of the ciliary abnormality, these pictures are present and they has to be investigated properly. So there are multiple problematic things: retinal degeneration, anosmia, because it is an embryonic origin. From the embryo, the ciliopathy started congenital heart defect, pancreatic cyst, truncal obesity, sterility, skeletal abnormality, limb polydactyly, renal abnormalities, liver disease, multiple disease, but there might be a combination of craniofacial or limb dactyly, polydactyly, or kidney disease together. There are multiple groups of different diseases, but whenever we do an ultrasound, whenever we see echogenic kidney, think about brain and encephalocele, think about polydactyly. If we see this type of things, then we can go towards a typical syndrome, then we can find out the particular gene which is affected, and they husband and wife are autosomal recessive. So ciliopathy refers to disease due to malfunctioning of cilia, singular is cilium. Cilia are organelles that are external extension of the cell membrane. Cilia fall into two main types: primary, which is immotile cilia, and motile cilia. Primary cilia are found in virtually every cell type, resulting in a wide range of anomalies, including central nervous system, eye, heart, kidney, liver, and skeletal abnormalities. These patients, these problematic babies, they take birth also. If we have, you can see a lot of blind, deafness, retinitis pigmentosa with kidney abnormality. They take birth and they have problem in their neonatal life and adult life. Why it is happening? So even if our pediatric friends are listening us, then we should find out the reason why. We should do a clinical exome sequencing to know the genetic disorder. So these are the different list of ciliopathy. Having azoospermia, married life 4.5 years with polycystic kidney, autosomal dominant. This is the pedigree chart, and andrological evaluation shows there is a obstructive, there is a congenital absence of vas difference. We did clinical exome sequencing, and you can find that there is a polycystic kidney disease, PKD1 gene in exon 15. This is autosomal dominant, and if they produce a child, there are chances that the 50% of the baby might be having the same disease without maybe a female or a male child, but they should have IVF with embryo selection for this gene by doing PGT-M, and they have undergone IVF, and we will do that. Now comes the RASopathy. What is RASopathy? RAS is a protein that is again this is a cell which is an antenna, gives a outside environment assessment and move or develop or orient related to each other with talking with each other through this cilia. Now, the cell is also having a cell membrane. This cell membrane is having a receptor. This receptor is for the ligand, like epidermal growth factor. It goes into epidermal growth factor receptor. With this receptor, there will be phosphorylation of the receptor. This phosphorylation which create this phosphorylation process by different steps to the nucleus. This is transmitted to the nuclear DNA machinery. So there will be transcription, leads to translation and formation of protein. Now this messenger which is happening inside the cell or cytoplasm is unique to every cell. While passing this message from the surface to the nucleus, It helps in secretion, proliferation, and other protein synthesis. Now, if any gene of different gene in between, different protein, I will name the proteins. Every protein is having a gene. If this gene is mutated and there is a backward flow toward the receptor to prevent proliferation, because the activation becomes inactivated, this protein gets inactivated by certain method, so that there will be no proliferation. Suppose this gene is mutated and this protein is produced in such a way that it doesn't inhibit the proliferation, then there will be cancer. That means this RAS and MAP kinase pathway is universal to every cell to have good secretion, controlled proliferation, producing a protein enzyme. So these genes are again conserved gene from beginning of the life in the universe. If there is a problem in the gene, there will be problem in the protein in this sequence. This protein will have abnormal action. If there is a problem with proliferation, hyperproliferation, cancer, or abnormal proliferation will create disease. So because all cell in the body are having the same system of RAS-MAP kinase protein synthesis, any problem with the protein will affect all cell. That means from the embryonic area, embryonic stage, if there is a problem, there will be abnormal embryo. This abnormal embryo might die or may grow abnormally. So there is no aneuploidy. The chromosome is okay, but the cell signaling is a problem in all cells. Because these cells are present throughout the body, so these genetic defect will affect all the body parts also. So there will be multiple organ abnormality. So these are the proteins which are required from growth factor. This is the cell membrane, and this is the RAS protein, KRAS protein, HRAS. These are different proteins which are in signal pathway, that is neurofibrin. If this protein is having abnormality, then there will be multiple organ problem which may create different sort of syndromic appearance. These syndromic appearance might be with Noonan, with Leopard syndrome, with lot of different problems in whole body will be created, and the baby will have multiple disorder in different organs of the body. So these are the genetic syndrome of RAS-MAP kinase pathway. Again, RAS-MAP kinase pathway is a system where the gene pathways are affected, the chromosome location is given, the protein produced by the gene, and different type of syndrome you can even this gene pathway is abnormal will create Noonan. SOS will create Noonan. So there will be genetic heterogeneity, and there will be a different gene affected creating a same type of abnormality, which is a Noonan syndrome. These syndromes which are present here are mostly autosomal dominant. Whenever there is an autosomal dominant disorders, usually these disorders are de novo disorders. Very few cases of autosomal dominant RASopathies are present in parents also, which might be has a very subtle variety. Very common is neurofibromatosis. So always think about this neurofibromatosis. Patient might be having only cafe ole spot, and the baby might be having some different problem with neurofibromatosis. So there will be difference of expression, different explicitity also. So there are these RAS genes, they create multiple disorders, and we should think about being, when we see the fetus and when we see this type of disease in sometimes in brain, think about problem in the other part, other organ also, like heart, like limbs, like kidney. When you make it together, then you can make it as a syndromic appearance. Then only you can go for this genetic analysis. RASopathies can be inherited, but they can be caused by spontaneous genetic changes. And most of the time, they are germline mutations. They are not somatic. So germline mutation might transfer, but most of the time, this is a de novo. In some cases, can run in families, 50% chance in patient parents passing addition on child. However, the genetic changes associated with most RASopathies are often sporadic. RASopathies include syndromes such as neurofibromatosis type 1, and there are multiple RASopathies which are, when we do an ultrasound, when we do prenatal ultrasound, we should think about all high or raised NTs are not aneuploidy. Whenever there is no aneuploidy, by doing QA-PCR and all, think about Noonan. And this is the basic pathway of the RAS MAP kinase pathway, and inheritance and genetic counseling of RASopathies will vary depending on the specific condition or gene. Gene testing of the affected individual and their parents is recommended, particularly as the phenotype spectrum is widely and mildly affected. Parents may not have been suspected clinically. I will come to a case today which we have done today with this RASopathy sort of thing. Patient has been sampled. Now we are doing this genetic analysis. These are the details of RASopathy. So plays an important role in cellular regulation. RASopathies result from dysregulation of the pathway due to constitutional gene variants. So always think about gene associated with the list, these RASopathies, and this is the basic pathway. By making different sort of protein deficiencies or abnormal mutation might create a Noonan syndrome, Noonan-like syndrome, CFC Noonan syndrome, Costello syndrome. These are all group of disorders which have different organ defects, but they appear together. So germline mutations, variant present in gamete, variation present in every cell in the body, including those of germline variants, can be passed to offspring. But somatic mutations are with they don't pass through the offspring. So, and then last is tubulinopathy. Tubulinopathy are complex microtubule. Tubulinopathies are mostly brain abnormalities, and these are due to the defect in tubulin genes, and most of the time they create this gene as autosomal dominant variant. Whenever there is an autosomal dominant matter and there is no problem in the parents, then mostly it is a de novo variety. Most of the time it is not recurring. So clinical characteristic tubulinopathies comprise wide range and overlapping range of brain malformation and clinical feature organized caused by pathogenic variants in gene encoding different isotypes of tubulin. So these are brain malformation. Lissencephaly ranges from thickened cortex. Various type of lissencephaly can happen. So there will be migration in the problem of neuron, and there will be formation in absence of sulci, and there will be thickened cortex and few shallow sulci. Whenever we do ultrasound of the fetal brain, always think about the sulci and gyri, which are very particular with the gestational age. If they are not appearing, if this Sylvian sulcus is not there, if it is smooth, think about there is some problem if you are doing an mid-trimester ultrasound. This is a case today we did. Patient was an IVF patient and she had date of exam is, the last period is 1st June, and we did embryo transfer. We did non-invasive prenatal diagnosis, prenatal screening, and you can see all 21, 18, 13 and 6 chromosome all are low risk. We were very sure that it is a low risk patient. But when we do an ultrasound yesterday, there is generalized anasarca, which looks like Turner, but because NIPT is normal, so we thought about that there is some other Noonan sort of syndrome also. There is a single umbilical artery, omphalocele. There is a problem of the heart, single ventricle, and I cannot do the full echocardiography, but there are problems with the heart. There is 4-chamber view is abnormal, and you can see
NT was also 5.
NT was 6.99 millimeter. Whenever this sort of thing happens, then because we did NIPT also, the NIPT was normal, low risk, then why it happened? So today patient was little bit disturbed, and this is the thickness of the neck area, and you can see the nuchal thickness, how big it is. This is the area of chest, the area of abdomen when we do, and this is the baby which is having generalized subcutaneous edema, and there is a problem with the 4-chamber view. We did Korean biopsy today, and we are doing this sample to understand what is the problem with the baby. And you can see the needle is going inside the placenta. And we did Korean biopsy today and waiting for the result. Because baby is not going to survive, so we did Korean biopsy also. We did amniotic fluid sampling, the amniocentesis also. And these are the cases which are so these are the cases where we should think about out of box, that it is not only the aneuploidy, it is not only the microarray. You have to go into the clinical exome sequencing method also to know about the defect in the fetus. I request Dr. Devashish to present his 2 ciliopathy problems. वो इसको वो